All right, everyone, welcome back from the morning break. I'm going to be taking over chairing this room for the rest of the afternoon. But to introduce myself, I'm Eleanor. I'm one of the producers at Oxford Global. Our next talk is from Anthony Schmitt, Senior Vice President of Science at Arima Genomics. Anthony Schmitt is the senior, well, as I say, he's the Senior Vice President at Arima. This is a San Diego-based biotech focused on advancing human health through the power of 3D genomics. Under his leadership, Arima has launched multiple products enabling life science researchers to enjoy the 3D genome using a range of applications, from genome assembly to translational cancer research and beyond. Prior to Arima, Anthony received his PhD from the University of California in San Diego, where he developed novel methodologies to understand mechanisms of gene regulations. Anthony is an accomplished scientist who has published his work in leading peer-reviewed scientific journals. Today, he's going to be speaking on a new frontier in biomarker discovery, how the 3D genome reveals novel disease mechanisms and therapeutic targets missed by other technologies. Thank you.
All right, thanks very much. How's the volume here in the back? It's good? Perfect. Okay, yeah, thanks for the introduction. My name's Anthony. I lead the science function at Arima. Over the past year, one of the things I've been working on pretty closely with our collaborators that I'm going to talk more about today is not just tech dev, but then the application of that tech in biomarker discovery. So I usually assume that folks have not heard of Arima. We're a pretty young company, born just, I guess, 6 years ago or so now. So just a little bit of background about Arima. Spun out of Bing Ren's lab at UCSD, where I did my PhD work. Shortly thereafter, 3 years thereafter, we launched our first product, and that's what I'll talk more about today, is sort of the underlying tech in that product, which is a sample prep method for measuring the 3D genome using next-generation sequencing. We've had a couple funding rounds since then as we've continued to grow. We've had 100 customer publications last year. We're a little over 300 now. And like she said in the introduction, serving really historically a base of discovery research applications from epigenetics and genome assembly, and now finding kind of a home in more translationally focused research, as well as some clinical work as well. Everything that you hear about today can be done yourself, either through kits, reagents, and software that we make available, or as a service. And I wanted to kick things off, I guess, with a story that's very close to the hearts of everybody at Arima, and it's also relevant for, I think, many reasons at this conference. It's really what I call our clinical origin story. And it started about June 2021. Had been in touch with Dr. Schmitt at NYU. This case, pediatric glioblastoma tumor in a 15-year-old girl that had recurred with sort of advanced disease growing very rapidly. They did a subtotal resection of this recurrent tumor and put it through the standard pathology workup at NYU, which is an FDA-cleared genome-packed assay, a ArcherDX-based RNA fusion assay. On this recurrent tumor, they couldn't find any drivers based on DNA or based on RNA. Here's a picture of that tumor there. So they had inconclusive results from the standard testing. And we had been in touch and basically sent our lab this FFPE block a week later. I put it on our mini-seq, got 10 million reads of genome-wide. Hi-C is the name of the technology. I won't go through how to interpret this particular plot, but what we found was a novel PD-L1 structural variant in this GBM tumor. So told that to Dr. Schmitt. He ordered a PD-L1 IHC, confirmed diffuse PD-L1 expression in this brain tumor. Shortly thereafter, a patient gets put on Keytruda, off-label, and then 9 months later, I stay in touch with the doctors on this patient, still has stable disease, out sort of enjoying life with her family. This was a really motivating story, I think, for us at ARIMA to really think hard about developing the 3D genomics technology here to identify structural variants that can impact patient management, like in this case. And you may be wondering, well, why didn't they just do PD-L1 staining in the very beginning? Well, the practice at NYU is they don't do PD-L1 staining because the diagnostic yield in pediatric brain tumors is so low that they don't do it. And they only did this because of the finding that we had from our analysis. And so what I hope you will walk away with today is, I have 3 goals. Assuming that you have never heard of 3D genomics before, you'll have an understanding of what I mean when I say 3D genomics, a conceptual understanding of how to leverage 3D genomics to detect structural variants, which I'm going to refer to as fusion biomarkers or gene fusions. And then lastly, a summary and some case studies or just sort of nuggets of data of how we've applied this technology to do clinical concordance studies with other technologies. But probably the most exciting thing is how we've applied this to cases like the one I showed you in the beginning that were driver negative based on sort of leading sequencing-based pathology assays or FISH, both in a prospective setting and in a retrospective setting. All right, so what is 3D genomics? Well, I like to position it relative to linear genomics, as that's what most people are used to thinking about. And most people think of the genome as just a 2 meter long stretch of A's, T's, C's, and G's, sort of shown here, wrapped around like this. And what 3D genomics is it's a sequencing-based sample prep technology where you still obtain the underlying sequence information of the genome. So it's a DNA-based sequencing assay. You get the same underlying sequence as conventional sequencing. But this information is obtained in the context of the 3-dimensional organization of DNA. So essentially, it's kind of like sequencing the structure or the 3D structure of chromosomes, where you get the sequence information and the structural information of chromosomes. And kind of sits sort of here at the center of this Venn diagram. Like I said, obtaining the sequence information, obtaining the structural information, which what I'm going to focus on today is how to sort of wield this tool to identify structural variants in cancer. And then for those who are more focused on, gene regulation, or if that's sort of part of your research interest, there's a huge body of literature that have used these types of approaches to understand gene regulation in 3D, link, regulatory elements, promoters, enhancers, and so forth. But I'll be focusing on this. I just have one sort of how the sausage is made type of slide that goes through very basic of what's sort of happening in the technology to capture that 3D genome information. So our technology commercializes a technique that you may have heard of called Hi-C. It stands for high throughput chromosome conformation capture, which was invented about a decade ago. It's really a glorified sample prep method or DNA extraction technique, and this is essentially how it works. You can take any sample, blood, cell lines, frozen tissue, formalin-fixed paraffin-embedded tissue, and if you look within the cells of these samples, the genome is not flat, it's not linear, but it's organized into a 3-dimensional space. Essentially what our technology does to take measurements of which pieces of DNA are close to each other, so kind of to obtain that structural information of the DNA within cells, is, well, right here in a formalin-fixed paraffin-embedded tissue, the cells are fixed. And so the DNA and the protein in the nucleus is all fixed, and you could have pieces of DNA that are spatially proximal to each other, like this light blue and this dark blue piece. So they could be derived from different chromosomes, they could be from the same chromosome but far away, and they've kind of folded back onto each other, as you can see in some of this drawings. All you're really doing is just cutting that DNA and then ligating it back together. So what you're effectively doing is creating these new molecules here that are now joined together that used to be spatially proximal. Now they're physically connected to each other, and your goal is really just to sequence these molecules When you map that back to the human genome, you could basically infer that this light blue piece and this dark blue piece were spatially proximal because they're linked in 3D. Okay. So at the very end of this, you just output DNA. That goes into library prep for sequencing. And then we have a containerized informatics pipeline on the back end. And what our collaborators and customers have really, I think, appreciated about this approach is that really this high-C portion just plugs into the front end of existing NGS workflows and ecosystem. The informatics is portable and scalable, easy to run, and it provides robust results. Before I get into some of those results, that's kind of like the underlying technology. And I think I just wanted to convey conceptually so you can think about how it has advantages and how it can be used to detect fusions in cancer and the advantages that it would have over other methodologies. So what's shown here on the left is basically a schematic of how gene fusions are detected with traditional DNA or RNA sequencing. So if you think about the genome in a linear fashion and you have a normal healthy cell, 2 different chromosomes, A and B. When you sequence those from a healthy cell, you get reads that pile up around gene A, reads that pile up around gene B. It's great. In a cell that has a gene fusion, let's say where A and B have fused together, if I only show you, and you sequence this sample, if I only show you the reads that are truly informative for detecting a fusion, they're going to be the ones that are split reads, where they sequence and they have a little bit of gene A in them and a little bit of gene B in them, and those reads right at the breakpoint is what cues you in to detecting a gene fusion with traditional sequencing. Now, I'm going to show you the same thing again, but now in the context of 3D genomics. So here on the left is a normal cell, and what's known about the biology of genome organization, which I'm not going to go into, so you just have to accept it as fact, is within an interphase nucleus, chromosomes tend to occupy their own space, and there's very, very little crosstalk between different chromosomes. That's a really important sort of foundational thing to understand in a healthy context. So here's B folded and interacting with itself, right? These little blue dots are where 2 things are spatially proximal on chromosome B, and these 2 pieces of DNA are spatially proximal on chromosome A. So in a healthy setting, from chromosome B, you have 3 long-range linkages. We call them 3D linkages. And on A, you have 2 linkages. Now in a cell with a gene fusion, right, now dark blue and light blue have fused together, those 3D linkages are no longer within a chromosome, but they're between 2 different chromosomes. And in this case, there's 4, right? And when I project this to sort of a linear schematic, you have your gene fusion here, where gene A and gene B have fused together, and here's those 4 linkages between chromosome A and chromosome B that you don't get in a healthy context, right? So this is sort of like inappropriate crosstalk between 2 different genes or 2 different chromosomes that don't normally happen in the healthy state. And this is just from one cell. So if I show you the same thing I showed you before, in a typical biopsy, whether it's from blood or solid tumor, there can be hundreds of cells, thousands of cells. So of course, this assay is done in a bulk setting. And so you sequence many cells, and you get many linkages between gene A and gene B across all these different cells. And what I want to kind of convey here again is in this schematic, you see, really, if you look right here at the breakpoint and you scan your eyes down, you'll get the reads that really overlap the breakpoint. But then the real advantage and the distinguishing feature from traditional sequencing are all of these reads that link chromosome A and chromosome B together. So we think of this kind of as like this amplification effect to help you detect fusions more sensitively, and it's DNA based. So our assay is also compatible with capture sequencing, where you can design probes to, let's say, capture the BCR gene. And this is from a sample, CML sample that had a BCR-ABL fusion. This is obviously a schematic. And what the technology gives here is reads that pile up at the breakpoint. So we did our targeted 3D genome assay using probes that target BCR. If you look at an IGV track, see all these split reads right there at the known BCR-ABL1 breakpoint. But because the BCR gene is going to be linked in 3D with those pieces of DNA in its sort of spatial neighborhood, you'll get all these linked reads between BCR and its fusion partner, ABL1, and the whole locus around ABL1. And what actual data looks like is over here on the very far right. These are all the reads that map to BCR. And every read that you see here is linked to another read that maps to ABL1 on a different chromosome. So that's depicting all these linkages. And there's actually over 7,000 reads that link these two genes together, but there's only 78 reads at the breakpoint. So there's basically a 100-fold enrichment of signal that helps you pinpoint that there's a fusion in a given sample. You might be wondering, well, what does a healthy sample look like? Of course, if there's no BCR-ABL fusion, there's no chimeric reads between ABL1 and BCR. Actually, pretty strikingly, there's no reads that link BCR or ABL1 in this entire locus. So there's effectively, in this case, the background is 0. And so hopefully that's a helpful conceptual understanding of how the technology can be used to identify fusions. We think about kind of 3 key advantages that I'm going to speak to in the final 10 minutes for using the technology to ultimately detect fusion biomarkers. These could be known fusions, they could be novel fusions, they could be fusions from archived blocks. We have projects going on actually right now with 15 to 20-year-old retrospective cases. And also finding fusions that have just been missed by other technologies. Okay, so in this final section, I'll talk about the clinical concordance data that we've generated, as well as the application of the technology to find fusions in undiagnosed or driver-negative tumors. So far, we've run approximately 220 patient specimens. Most of these are retrospective. Most of these are solid tumors in FFP format. You can see the breakdown of indication here, with the majority of them being CNS or sarcomas, different types of sarcomas, but I'll talk about that more in a second. But also lung, some of the solid heme indications, GI, myeloma, et cetera. So 20 different tumor types, because we often get asked, well, does the technology work in tumor type X? And this is our experience so far across 20 different tumors. And of this 217, 81 of those were for clinical concordance with FISH or RNA panels or DNA panels, and the rest were driver-negative tumors. So I'll go through this quickly because it's not super exciting, but every sample that we've looked at that has had a known fusion based on other technologies like Archer or FISH, karyotyping, et cetera, we've been able to find. And this is true across many different indications. This was actually also presented this past year at AACR with scripts where a variety of different orthogonal techniques had been used and found various fusions. A lot of these are lung specimens, so there's a lot of ALK fusions, also NTRK fusions. You can see here that our technology, because it has those sort of linkages between genes, it inherently just captures both genes in the fusion, whereas something like break-apart FISH obviously is only detecting one of those genes, so it detects the fusion partner and is concordant with the other methods. Same story here with Children's Mercy. This was also presented at AACR this past year. What I wanted to point out here was just the archival period of these alveolar rhabdomyosarcomas in the 8 to 12 year range, and the input DNA can also sometimes be very low from these older specimens. And this is how much we used into our library prep over here. All right, so let's get back to this case as sort of an introduction to the driver-negative data that I'm going to show you in a second. What we realized sort of early on as we began to sort of explore the science of these driver-negative cases is looking at other publications that have estimated the percentage of tumors that are driver negative after DNA and RNA sequencing, like this study from MSK Impact. The driver negative rate shown here in blue in these stacked bar plots varies per indication, but as a rough estimate, about 450 or 500,000 driver negative advanced cancers per year in the US alone. So we felt this was a pretty important application and problem to solve with our technology. And so here's some of the data. We've analyzed 140 driver negative cases so far. So these are samples that have come to us that were already tested by NYU's panels or MSK Impact. So 140. We call our fusions, and then what we found is that in 46 tumors, or 33% of all the samples that we analyzed, there's a fusion that's associated with a biomarker that is the target of an FDA-approved therapy today. In 6% of them, the biomarker is targeted by a therapy in an ongoing trial. And in 14% of them, the biomarker with the fusion that we detected is of prognostic, diagnostic, or pathognomonic significance according to the NCCN biomarker compendium or OncoKB. So I should mention that pretty much our reference database to attribute the significance of the findings is all the NCCN biomarker compendium, World Health Organization, or OncoKB. And then in the final 17%, these are biomarkers associated with genes of a role in cancer, but not necessarily in these top three tiers. And most of this data is being driven by just a few indications. So we have 38 gynecological sarcoma tumors in which the sum of what we call tier 1 or level 1, level 2, and level 3 evidence is about 68%. The solid hemes is also 68%. The CNS tumors is a little low. And this is a pretty interesting cohort that we're developing right now. These are colon tumors that are driver negative, but they were previously sequenced at Caris. And so we're getting them post-Caris where there's been no actionable finding in the Caris report. And so far in these 5 CRCs, we can find a tier 1, 2, or 3 biomarker in 4 out of the 5. And we're working to build that up now. Here are some of the markers themselves. You can see the ones in purple are the ones that are targeted by an FDA-approved therapy. So the RAD51s would be for PARP inhibitors. You can see NTRK 1, 2, and 3 here. The ones in light blue are the targets of the drugs in trial, and the ones in black are the ones that are diagnostic or prognostic in the correct indication. So a PLAG1 fusion in a uterine sarcoma. Right? And to really kind of take a deep dive into any of the specific genes or any of the specific indications, we could talk about that offline because there won't be time here. And I want to end with just one example of another prospective case, which was a 37-year-old female with diffuse infiltrating glioma, presented with onset seizures and a 2-centimeter mass in the temporal lobe. Tumor was resected. Biopsy was analyzed again by NYU. Their genome packed assay, negative. Fusion seeker, negative. They even actually have a brain tumor methylation classifier at NYU that diagnoses brain tumor subtypes, and this actually was almost a perfect match for a low-grade glioma driven by MyB or MyBL1. But the conundrum here was there was no MyB or MyBL1 fusion detected, even though MyB and MyBL1 are targeted in the NYU fusion seeker panel, right? And based on the array data, there's also no CNVs, so pretty quiet genome from a structural perspective. But anyway, the tumor was sent to us. We found the MyBL1 fusion here. This is actually a copy-neutral reciprocal fusion based on the pattern here, which I don't have to go into, but you can see there's a breakpoint right here in the MyBL1 gene and a breakpoint right here in the MAML2 gene. So this was actually a very favorable prognosis, and what it meant for the patient after this was validated was no adjuvant therapy after resection. So we've had 4 prospective cases total. A glioma that I told you about in the beginning with PD-L1. The patient was put on pembro and has had stable disease for the past 9 months. We also have a triple negative breast that I didn't have a chance to talk about, also with a PD-L1 rearrangement. Patient was put on pembro, stable disease for the past 6 months. An NTRK fusion, put on larotrectinib, and then the MyBL1 case that I just showed you. So in conclusion, hopefully made you aware of 3D genomics and what it can be used for. It's concordant with other approaches. Also works back in time with our archival specimens. You can find fusions that are not being detected by other NGS-based tech, both in a prospective setting, like the off-label use of pembro, and in a retrospective setting where most of our data comes from. I just want to acknowledge the people who enabled this all to happen, mainly Matthias Nuderl at NYU, Darren Siegel at Scripps, Midhat Faruqi at CMH, and then Sarah Chang at Sloan Kettering. Thanks.
Great, thank you, Anthony. I think we have a question already over here.
Really interesting presentation and platform. So of course you focus on the gene fusions, and there's really nice application of that, but I was curious about an analysis where you're comparing like the relationship between genes physically in space and any correlations with like prognosis or response or anything like that.
When you say physically in space, you mean like proximity to the nuclear periphery or interior?
Or just like in the 3D, finding like, I don't know, P53 super close to, I don't know, like ALK or something, and seeing any correlations like in that 3D space?
Yeah, so far we've just been focused on the fusions, not necessarily just inherent 3D proximity that's not associated with a fusion. So, I don't know.
Okay, because it just made me think of when you're thinking about like enhancers for genes and how enhancers work at such a distance, but physically 3D, if it's close to the promoter of another gene, you can think about how it changed gene expression.
Yeah, absolutely. I mean, one of the things that I didn't get a chance to talk about, but we're really, really keen to look at, and you can with our technology, is whenever you have a fusion, you're also changing the 3D interaction landscape on that rearranged allele. And there's been actually a handful of papers, some that we're co-authors on, that specifically study the consequence of a fusion in terms of the regulation of the genes around that fusion, because now it's kind of put into a sort of a new gene regulatory environment that it's not typically in. And we have some papers that I cut from this just because of time, where that's led to some really interesting things. Maybe we can talk about it after. Yeah.
Yeah, very interesting technology. Maybe a related question. So at the moment, does it have the potential to fully replace karyotyping? I mean, because then it's more just fusion, right? So you have inversion, translocation, whatever, structural variants. Yeah.
Yeah. The one case that comes to mind, and we can talk about it more afterwards, is we have collaborators who specialize in things like lymphoma, where there's a lot of interest, or other liquid cancers, where there's a lot of interest in sort of seeing this as like a highly multiplexed type of FISH assay, but it's also sequencing based, so you can get copy number information at the same time. So like in one shot, you can look for all fusions at a genome-wide scale or at a targeted scale, plus the inherent copy information just from the 1D sequencing coverage.
Hi, great talk. Just a quick question. You've shown concordance with the other technologies. Can you comment as to whether or not there have been instances where the other technologies are unable to detect and you have a positive result?
Yeah, I guess that's kind of the inverse of all of the driver negative data that I showed. All of those findings were not detected by the previous methods. Like the MyBL1 fusion was targeted by the Archer panel at NYU, didn't find it, and then we found it. So I guess that's kind of the inverse way of answering the question, but yeah.
I think that's all we have time for in terms of questions, but feel free to approach Arima Genomics at their booth in the exhibition hall if you have further ones. As for our next two presentations, unfortunately, they won't be taking place. Kenneth Ramos's presentation, which was meant to be at 12:45, we are arranging for that to be recorded and put onto our swap card platform within the week. So if you would like to hear that, then that's that. Otherwise, we'll be back in here at 2:20 for a presentation with Jeffrey Wallen. There are other presentations going on in rooms 1, 3, and 4, though, for your interest. Thank you.